Hey everyone, welcome to The Explainer. Today we are embarking on a really fascinating journey into one of the most incredible architectural feats in all of biology, the eukaryotic genome. We usually think of DNA simply as a code, right? Just a string of letters detailing the instructions for life. But today, we're completely shifting our perspective. We're going to look at DNA as a physical object. It is a structural marvel that requires an astonishing level of packaging and organization. And this isn't just abstract science. This precise architectural packaging is exactly how a single fertilized egg flawlessly develops into a complex human being. And when this packaging goes wrong, it's often the root of diseases like cancer. So, let's just get right to the massive physical dilemma here. Picture this. If you were to take a single human cell and stretch out its DNA, it would be roughly 2 meters long. Now, you have to pack that entire 2-meter thread into a cell's nucleus, which is only about 6 micrometers in diameter. To put that into perspective, that's basically the equivalent of trying to pack over 20 miles of extremely fine thread into a single tennis ball. And you have to do it without a single tangle, while keeping the thread perfectly accessible so it can be read at any given moment. How on earth is that even physically possible?
To answer that, we're going to break this explainer down into 5 stages. First, the DNA packaging problem. Then, building the 30-nanometer fiber and the mitotic chromosome, before exploring the 3D nuclear architecture. And finally, we'll wrap up by comparing eukaryotic and prokaryotic genes. All right, section 1, the DNA packaging problem, compressing the code. Let's get right down to the foundation. So, nature doesn't just jam all this DNA in at once. It uses a super elegant, stepwise, layered approach to compaction. Starting way down at the 2.5 nanometer double helix, at the very top, we don't just jump directly to a massive 1400 nanometer mitotic chromosome. The DNA is spooled, those spools are coiled, and those coils are looped into increasingly larger structures. It is an absolute masterpiece of biological engineering. The secret to this entire hierarchy begins with something called the nucleosome. This is the absolute fundamental unit of chromatin. Think of it as a microscopic protein spool. You've got roughly 147 base pairs, those are the rungs of the DNA ladder, wrapped really tightly around a core complex of 8 histone proteins, specifically 2 each of H2A, H2B, H3, and H4. This is the first essential step in our packaging problem. By wrapping the DNA around these histones right out of the gate, the cell creates a compact, manageable unit, preventing tangles from the very start. And we can actually see this when scientists look at chromatin under an electron microscope at low ionic strength. It forms what they wonderfully describe as the beads on a string formation. I mean, it literally looks exactly like that. Each of those tiny dark beads is a nucleosome, and they're connected by a string of linker DNA. This isn't just an abstract theory. This is the observable 11-nanometer structural reality of our genetic foundation. Moving on to section 2, building the 30-nanometer fiber, higher order compaction. So to fit those 2 meters of DNA into the nucleus, those 11-nanometer beads on a string need to fold up even more. But here's the wild part. Structural biologists actually still debate exactly how this next level of folding happens. As the chain compacts into a 30-nanometer fiber, 2 primary competing models emerge, the solenoid model and the zigzag model. Let's break these down. In the solenoid model, consecutive nucleosomes coil neatly into a helix, forming a central cavity, kind of like a hollow spring, with about 6 nucleosomes making up a single turn of the coil. On the other hand, the zigzag model suggests that the nucleosome beads crisscross back and forth, with a linker DNA passing right through the central axis, resulting in a denser, interwoven column. Either way, they both solve the exact same problem, taking that linear nucleosome chain and drastically shrinking its physical footprint. Okay, section 3, the mitotic chromosome, ultimate compaction. For most of a cell's life, chromatin is actually just moderately condensed. But when it's time for the cell to divide, it needs the ultimate level of compaction. Enter the mitotic chromosome. This is that iconic X shape sitting at a massive 1400 nanometers wide. A couple of key landmarks stand out here. At the very ends, you've got telomeres. These are repetitive sequences that act kind of like the plastic aglets on the ends of your shoelaces. They protect the chromosome ends from degrading or fusing together. And right in the middle, holding the 2 sister chromatids together, is the centromere. This is absolutely essential for making sure the chromosomes are evenly segregated when the cell finally divides. But here is the really crucial point. This packaging, it's not just about saving space. It is literally the primary way a cell controls its genes. We categorize chromatin into two distinct types based on how tightly it's packed. You have euchromatin, which is loosely packed, meaning it's physically accessible to the cell's machinery, so it is transcriptionally active. Then you have heterochromatin, which is densely packed and tightly spooled away. Because the molecular machinery physically cannot reach the DNA sequence, heterochromatin is transcriptionally silent. Basically, structure dictates function.
Which brings us to Section 4, 3D nuclear architecture, a molecular city.
So we've looked at the individual threads. Let's zoom out to the whole nucleus. And let me tell you, it is not just a chaotic bowl of molecular spaghetti. When we visually map the nucleus in 3D, we see that every single chromosome occupies its own distinct, non-overlapping space. We call these chromosome territories. This intense spatial organization is a massive factor in influencing gene expression. Where a chromosome physically sits within that 3D space directly dictates how easily its active genes can be read.
And just like a bustling city has industrial, financial, and residential districts, the nucleus has localized neighborhoods dedicated to specific tasks. We call them nuclear bodies. You've got the nucleolus, which handles ribosomal RNA synthesis, along with perispeckles, processing bodies, Cajal bodies, and nuclear speckles.
To understand how these neighborhoods work together, let's compare two closely related ones, Cajal bodies and nuclear speckles. You can think of Cajal bodies as the manufacturing plants. Their main job is building and modifying SNP RNPs, small nuclear ribonuclear proteins. These are the vital components of the spliceosome, the machinery that actively edits our RNA. Nuclear speckles, on the other hand, are like the distribution warehouses. Located right next to actively transcribed genes, they act as storage and delivery sites, constantly supplying those splicing factors manufactured in the Cajal bodies to the genes that need them. It is a completely synchronized, highly coordinated logistical network.
All right, final section, Section 5, eukaryotic versus prokaryotic genes. Comparing gene layouts. Let's pivot from the macro 3D architecture down to the linear DNA sequence itself. When we compare our genes to the simpler genomes of bacteria, the evolutionary split is just staggering. Eukaryotic genes are sprawling and incredibly complex. They are filled with non-coding segments called introns that interrupt the actual coding exons. Plus, they have an intricate array of upstream and downstream regulatory sequences. But look at the prokaryotic gene structure. It is heavily streamlined. Multiple different genes, gene A, B, C, D, E, are all clustered together into a single unit called an operon, and they're all controlled by just one promoter. Breaking this down, the divergence is stark. Eukaryotes utilize a complex system where individual genes have dedicated, highly sophisticated promoters. They use enhancers, silencers, and those intervening introns that require that massive, complex splicing machinery we just talked about. Prokaryotes, though, prioritize raw efficiency. They lack introns entirely. And by grouping multiple related genes into operons, one single genetic on-off switch can activate an entire metabolic pathway all at once.
So, throughout this explainer, we've looked at the sheer architectural brilliance of the eukaryotic genome, from a tiny 2.5 nanometer double helix wrapping around histone spools, folding into fibers, condensing into distinct chromosomal territories, and operating alongside complex nuclear bodies. But here is the most mind-blowing part. This incredibly rigid, organized hardware, it's not static. It is highly dynamic. It is constantly shifting, loosening, and tightening right this very second, depending on exactly what your cells need. Which leaves us with one massive final thought. If everything we've just looked at, the nucleosomes, the 30-nanometer fibers, the territories, is the physical hardware of the genome, what controls it? Well, the answer lies in the epigenetic software. Chemical tags like DNA methylation and dynamic modifications to those histone spools create a complex code that actively manipulates this massive physical structure. This epigenetic layer opens and closes the architecture in real time. But exactly how that chemical software rewrites our physical biology without ever changing the underlying genetic code, well, that is a profound mystery we'll have to explore in another explainer. Thank you for joining me on this incredible journey through the architect's blueprint of life.